Hello everyone, welcome back to our discussion on Mendelian genetics. We were discussing about the Mendelian laws of genetics. In the last session, we saw the two laws of Mendelian inheritance, and that was the law of unit factors and the law of dominance. And now we are going to discuss the third law, which is considered as the first law by some of the authors, and this is considered as the first law of Mendelian inheritance. And this was proposed on the basis of the monohybrid as well as the dihybrid process. And here what we are going to say is that, so we'll see and then discuss this particular. So we are going to discuss the third law of Mendel inheritance, which is the law of segregation. So what Mendel says is that if we take, so we are going to consider a monohybrid cross, which is the same flower color, the F parent one with red and white flowers. So we know the gametes will be, what happens during gamete formation is that these two are the factors which were considered by Mendel. They segregate during gametogenesis, they segregate and separate into two different cells. As a result, you get a single type of, the same thing happens for this after meiosis. You get the gametes, and in F1 you get capital R, small r. Okay, and the same thing during, or if this is the F1 and F1 cross F1, you get the gametes. I'm not drawing the entire gametes here, and smaller. And the next generation will be, you get when you cross this, you get capital R, capital R, smaller, capital R, smaller, and smaller, smaller. So this is a normal Mendelian monohybrid cross. And here what we can see is that in the F1 generation, as we said, so this was the white colored flower and the other one is the red color flower. And in the F1 generation, you do not see the white flower color. And this white flower or white colored flowers, they appear in the F2 generation. In the F2 generation, you can see this white flower colors. And what Mendel assumed is that this particular factor which is responsible for producing the white flower color in the F1 heterozygote, this particular allele or this particular factor which is responsible for producing the white flower color is not getting mixed, or it does not blend during gamete formation in the F1 generation. So what we are going to see is that if you take, if we consider this as, so we are going to consider the F1 cross F1 here. So if we consider this, you have two Mendelian factors for F1 like this. One is capital R, the other one is small r cross. This one which is capital R, and the small one which is small r. So these are the factors which are being considered by Mendel. He considered these trait determining units as factors, so I am drawing it as factors. So the same thing, the same what we saw here happens. So what he says that, so this was parents of the second generation, and when gamete formation happens, what he says that now we know the process of meiosis, the different process of meiosis as we discussed in the introductory session, what happens during meiosis for homologous chromosomes, etc. Homologous chromosomes, they separate. As a result, in gametes we have half the number of chromosome, etc. Is the information that we have now. But when Mendel proposed this particular law of segregation or Mendelian laws, he did not have any idea about meiosis. what happens during meiosis, or what is happening during gamete formation, whether the chromosome number, the idea about chromosome, or what is a chromosome itself was not known during his period. So he didn't know about what is meiosis, etc. So what he considered, what now I'm going to explain what Mendel considered in such cases. So what Mendel considered was that according to earlier beliefs, that means what we discussed in the introductory session of Mendelian laws, that means they considered these hereditary determining factors as fluids, or they where the consistency of that particular factor was fluid, and as a result they will mix up or they will blend. So if it was a fluid, so consider if it is a fluid, what happens is that if these particular factors had a structure or had a was in the form of a fluid, what is expected is that when these form gametes, or when they combine, when R and small r, that individual is formed, his gametes will have a mixture of this. Mixture in the sense, if it was a fluid, I'm taking a condition hypothesis, when whether if it is a fluid as hypothesized by the precursors. So if it was a fluid, what you will get is that these two will combine. Because it is fluid, it will mix up, it will blend. As a result, they will combine and produce a single gamete, which is capital R small r. It will be combined. Because you cannot see this, or you cannot expect this in the F1, because in F1, because of dominance, you are not seeing small r, whether small r is present in the blended form or separate form. Whatever the case may be, in F1 you cannot prove whether it is a factor, or whether this R, this small r is a factor which is blending with capital R. Whether it is seen blending or whether it is combining with small r cannot be proved in F1. Because in F1 we are getting a result where the dominant trait is shown. Okay, what I am trying to explain is that when Mendel did his experiments, he didn't have any idea about this genotype, etc. Okay, what was observed by Mendel was just the phenotypes. From the phenotypes, he started to deduce the genotype. So what he sees is only the phenotype. So what happens in the F1 generation in this particular case is that in the F1, even if it is in the form of a fluid, even if it mixes, because of the law of dominance, you can see red color flower without any doubt, because of the law of dominance. But if it is blending, if blending occurs, consider this particular factors in the form of fluid and consider they blend, and as a result of blending, the small r becomes inactive, or small r does not have any, or after when two fluids get mixed up, you cannot separate it further. If it is discrete particles, consider you are having two balls, and consider you are having two colored jellies. And if the two color balls, you can separate it. Even though you stick it with some glue or something, you can separate it somehow, right? But if it is a jelly which is of two color, red and colorless jelly, you mix it thoroughly. It is you cannot easily separate those jelly into again white and red. Is that clear? So what Mendel tried to explain is that if the factors were in the form of fluids, if they didn't have any discrete shape, what will happen is that in the F2 generation, you will not get a white flower color because it got mixed up. Now R is mixed with red. You cannot separate small r from capital R if it is fluid. But if it is discrete particle, what can happen is that discrete particles can be separated. During gamete formation, discrete particle can be separated. Now we know that the discrete particles, which are chromosomes, are separated because of meiosis or gametogenesis. But what Mendel said was that if these particles are discrete like this, so we are now erasing all this, mixing up fluid, etc. Now we consider them as discrete particles itself. If these are discrete particles inside the cell, you can separate them into two different cells or into two different gametes during gamete formation. So what happens here is this capital R will be separated and small r will be separated. We know that it happens in anaphase now, and this also can be separated. And because they are separated, okay, and this separation is what is called as segregation. That means the alleles or the factors that control a particular character is, or they can segregate into two different gametes. Okay, that is what is called as law of segregation. That is this particular event. That is, because these are separate entities, they can be segregated into two different gametes, or they segregate into two different gametes during gamete formation. And this is what is called as law of segregation. And because of, and law of segregation was proved only because you get, so these are the offsprings which are formed. This is proved only because you get this particular result here. If this was not, or this white flower or white flower plant did not appear in the F2 generation, we could not explain this particles as discrete structure because it is getting disappeared. But that is not getting disappeared, it is not getting blended with capital R. It, even though in the heterozygote, it is keeping, or it is having its individuality, or it is having its distinctiveness. Is this clear? So this is what is called as law of segregation. That is, in simple terms, I explained all this just to put before you what Mendel has assumed in this particular experiment. And the particular takeaway point in this particular law of segregation is that during gamete formation, the two factors which are responsible for controlling a trait is not mixing up or blending together. They can be segregated into two different gametes. And this segregation, or this particular property, is what is called as the law of segregation. So we'll come to the explanations which is given here, which will make it more clear. So when a pair of contrasting characters, so here the Mendel's first law of inheritance states that the two factors, which we now call it as the alleles of a character, they do not mix up or blend together during gamete formation because they are distinct and separate particles which are called as unit factors. They do not blend. And as we saw, what happens here is during gamete formation, this you have capital R and small r. During gamete formation, this capital R and the small r segregates into different gametes. And thus, each offspring will receive only one factor from its parent. So that's clear. That is, this is one parent and this is another parent. And in an offspring, this R is one parent from one parent and this is from another parent. So what is shown here is, thus each offspring will be receiving one factor or one allele from this and the other allele from. So in every case, the same thing will happen. And the alleles do not mix up or contaminate during gamete formation and they remain pure. This is another term to be noted. They are remaining pure and containing discrete factors which can be either dominant or recessive. It does not matter whether it is a dominant allele or recessive allele. They will remain pure inside the, without mixing up. That is what is called as pure. And hence this is also called as the law of purity of gametes. And this is this was proved by the, as we said earlier, by the reappearance of the white flower color in the F2 generation. So if there was blending or mixing in the hybrids, when in the hybrids, if it was as I said earlier, in this particular capital R small r, if there was a mixing, or capital P small p, whatever it is, in the heterozygote, if there was a mixing happening or blending happening in them, this white flower color will not have appeared. Because white flower colored appeared in the F2 generation, it is clear that the gametes are not mixing up, they are retaining their individuality and they segregate into different gametes. And because each of the factors they remain pure without blending, this law is also called as a law of purity of gametes. Is this clear? So by this particular law of segregation, Mendel tried to prove two things. One is the law of, or he was reproving the law of law of unit factors, in the sense that because there is, he proved that there is no blending, and because there is no blending, it is for sure that, or it can be proposed that these particular factors, they are remaining as discrete molecules. And because they remain as discrete molecules, they segregate during gamete formation into two different cells. So if you have a hybrid capital R small r, capital R is because they according to the meiotic principle, as we know now, this capital R, these are homologous chromosomes. Capital R will be separated, this chromosome will be separated and this will be separated. As a result, they move into two different gametes, and this gives rise to two cells. Is normal meiosis. So Mendel did not consider them as chromosome. Mendel just considered them as discrete spheres. These discrete spheres, they are segregating into two different gametes, and that was proved by this law of segregation or law of purity of gametes. So this was the third law of Mendelian or Mendelian law of genetics. And now we are moving on to the last one, which is the law of independent assortment. So to state simply, or in one line, you can define this law of independent assortment in the sense that it states that the alleles of separate traits or characters are independent of one another during gamete formation. That is the one line explanation of what is called as law of independent assortment. So stated, or this particular law is stated clearly in this particular single sentence. So we'll look on to that one once more. It states that the alleles of separate traits or characters. So separate traits means it might be a dihybrid cross where we have two different characters or traits, and you have two different alleles for that. So in such a case, what independent assortment say is that these alleles of separate traits or characters are independent of one another during gamete formation. Okay, so we'll explain this and come to the description again. So this is what is called as the law of independent assortment. So what was stated was that the alleles of two different characters. So you have two different characters here. One is seed color and the other one is seed shape. Here you can see this is the dominant for seed shape, capital R, which is for round, and the other one is for small r is for the recessive one, which is wrinkled, round wrinkled, and you have the dominant one color which is yellow and the recessive color which is green. And what we are going to see here is these particular alleles, that is capital R and small r. So we are stating it, we are taking two alleles separately. Capital R and small r are one pair of alleles, and you have capital Y and small y. These are two separate alleles which determine two separate characters. And what independent assortment say is that the alleles of two separate characters. So if I take capital R here, and you have two alleles, capital Y and small y. Okay, so what you say here is This capital R, which is an allele of a particular trait, it is independent of, or it is independent in the sense that it can assort either with capital Y or it can go with small y during gamete formation. When gametes are formed, there is no set rule that the dominant allele should go with another dominant allele. Is that clear? So what we are trying to say here is, this is a dominant allele, capital R, and it has chance to combine with either a dominant allele or a recessive allele. And this combining, or this is entirely a chance. It can either go with capital Y or either it can combine with small y during gamete formation. In the sense that there is no set rule that the dominant allele should pair only with dominant and dominant should not pair with recessive alleles. So this independency where this independency of an allele of a particular trait to combine with all the alleles of another trait is called as independent assortment. So we'll state it clearly once more. So what we try to say is that when gametes are formed, we'll take the same example here. You have the F1 heterozygote, capital R, small r, capital Y, small y. When such an individual forms, so this is a homologous pair and this is another homologous pair, like our first chromosome and second chromosome. Is that clear? We have 23 pairs of chromosome, okay, and that makes up 46, of which consider this is our first pair and this is our second pair, that is first chromosome and second chromosome. So what should happen during gamete formation is that you should have the contribution of the first chromosome and you should have the contribution of the second chromosome. Then only the gamete will be complete, otherwise it will be defective. If you take the example of human chromosomes, in the case with the human chromosome, in a human gamete, it should have the contribution from all the 23 chromosomes. Okay, in the gamete forming cells you had, or the parent cell you had, if the parent cell, it was 46 chromosome, which was 23 and 23 pairs. Okay, when a gamete is formed, it should have 23 or representatives of all the 23 in all the 4 gametes after meiosis. The same thing is applicable here. That is, when a gamete is formed, the gamete should contain the allele for round as well as the allele for yellow. Okay, when such a gamete is formed, there is no set rule that the dominant should pair only with a dominant trait. These are two different characters, and there is no rule that the dominant one should pair, or the dominant one should be present only with the dominant trait of the other. It is a chance, and the chance is nothing but, as we said during meiosis, it is just how they align during a metaphase of meiosis. Okay, and depending upon that, so what Mendel thought was that you have 4 discrete particles like this. See. Okay, we take it once more. So this was the heterozygote, and Mendel considered that this is small r and the other one I take it in different color like this. And during gamete formation, Mendel considered that when they form gametes, or during gamete formation, so what we'll see what happens during gamete formation here. This is what we call it as the metaphase arrangement, and during metaphase arrangement, So this is capital R, and this to show this is capital Y. Capital Y. So what Mendel said was that during gamete formation, when gametes are formed, so this can be, I mark it as small r here and small y here. So what Mendel suggested in this law of independent assortment is that this alleles, they can combine, or this is one type of arrangement where where these two get separated into this pole and these two get separated into this pole. As a result, now we are having capital R and capital Y in one cell and small r and small y in other cell. This is one type of arrangement. Mendel also considered that they can also align in a different manner, in the sense this can be small y and this can be capital Y. That means this capital R has an independency to assort with either capital Y or small y. And that independency will result in the generation of 4 different gametes as shown here. As you see here, from this particular parent you have 4 different gametes, and here you can see the independency where capital R is pairing with, or it is assorting with, it is not pairing, it is pairing is actually a wrong term. It is assorting with capital Y, and capital R is assorting with small y. The same is the case with small r. Small r is assorting with capital Y and small r can assort with small y. The same if you, I mentioned it with capital R and small r, the same if you take with capital Y and small y, the same thing happens. So you get 4 different types of gametes in this particular case, and this is due to what is called as law of independent assortment. So to make the points clear, so the law of independent assortment states that the alleles of separate traits, separate in the sense, what separate here means that one is seed color and the other one is seed shape, are independent of one another during gamete formation. They do not have any condition where the dominant allele will assort with the dominant allele, or recessive allele only assorts with a recessive allele. So one factor or allele of a particular gene, for say seed shape, is independent to assort with any of the 2 factors. That is what we said. That is, capital R is having the independency to assort with any of the 2 alleles. That means either capital Y or small y. So what happens, the example is illustrated here. In the dihybrid cross, the dominant allele for seed shape, which is capital R, can randomly assort with either capital Y or small y, forming the gametes capital R, small y is capital R, capital Y, and capital R, small y. The same is applicable with the recessive allele, which will give you small r, small y, and small r, capital Y gametes. This is how you get, or this is how we are getting 4 alleles, or sorry, 4 gametes from your F1 heterozygote. And this is due to the 2 possible metaphase arrangement of homologous chromosome, or it can be due to the equal chance of any of the members of the pair to go with, or to go to any of the poles. So all the 4 types, F is missing there, all the 4 types of gametes produced will have the equal frequency, and that is what is shown here. It is having equal frequency in the sense each of the gamete will be formed in the chance for forming is equal, which is 25% 1/4. So they will be formed in equal frequency. So this is what is called as law of independent assortment. To state in simple terms, you can say that a particular allele of a particular trait is independent to assort with any of the allele of another trait. Is that clear? If you have 2 different characters or 2 different traits, one allele of a particular trait is independent to assort with any of the allele of the other trait. Okay, that is, you just keep this in mind. The capital R allele can pair with either capital Y or small y. That is what is called as, and this independency, that is capital R can go either with capital Y or small y. That is what is called as the law of independent assortment. So this, or with this, we come to the end of the Mendelian laws. So we have discussed all the four laws of Mendel: the law of unit characters, the law of dominance, law of segregation, which we stated that the gametes remain pure, they do not blend. As a result, they segregate during gamete formation, and that is why you get the recessive trait in the F1 generation. Is that clear? And coming to the independent assortment, as we said, the allele of a particular trait is independent to combine with or assort with any of the allele of another trait. Okay, so this is the four laws of Mendelian genetics, or which was proposed by Mendel. With this, we complete the Mendelian laws. The next session will be about some special type of crosses like back cross, reciprocal cross, and test cross, etc. We will discuss that in the next session. Thanks for listening.